Good afternoon, everyone. I'm going to start today's class. So I think my screen is visible. Uh, okay, so let's start today's class. So in this class, in this week, Sir has taught about the POCT devices, then the cellular uptake of the nanomaterials. Then he talks about the cell death and cell assay. And lastly, he talks about the uh, nanotoxicology, the effect of nanomaterials on the cells. So we'll go topic by topic. Okay. So first one is the what is POCT devices? So you already know that POCT devices is the point of care testing devices. And this is a kind of medical devices which you can use by your own. It is very easy to use. They are frugal and they are portable, right? And they can give result very rapidly. So it is written here that these are medical devices that perform diagnostic tests near the patient, providing rapid on-the-spot results instead of requiring a sensor lab. Generally, for the blood test, for glucose test, for diabetes test, we go to lab, right? Any blood parameter. But if we have the POCT devices, like glucometer is one kind of a POCT devices, pregnancy test kit is one kind of a POCT device. So that is very handy. We can use on our own, and it gives 95% accurate results, right? So this is the advantage of using POCT device, right? So we'll see some examples. That is the urine test strips is the POCT devices, TB test strip, glucometer. Every almost we know about these common POCT devices. HIV test kit, heart rate monitor, pregnancy test kit, right? So you can see that it is the bedside testing. This is a handheld device. Okay, you don't need a big instrument, you don't need to separate room for that. Okay, this is very handy. It also prevents emergencies. It gives quick results and it saves your time. And this is the project of World Health Organization also. So they comes in this compact form, assured. This is affordable. These devices are affordable. They are sensitive. They are specific. They are user friendly. They are robust. They are equipment free. That means you don't need a large equipment or gold standard equipment. That is deliverable. They are also portable. Okay. So this is the POCT devices or handheld devices. Next, what is the paper-based diagnostics? So, paper-based diagnostics are low-cost, disposable, microfluidic devices. So, all these are paper-based diagnostic devices are part of this POCT devices. Okay, we use simple paper to make the device. So it is low cost, disposable. That means once you use one paper strip, then you can remove that. Then you can dispose that. So this is the low cost, disposable microfluidic devices, often called microfluidic paper-based analytical devices that use paper as a substrate for point of care tests and sensors to detect various molecules, pathogens, and environmental contaminants. Right? So here they use paper. So you can see here that With the paper also we can do the colorimetric sensor. That means the color change we can see with the help of this paper. The chemiluminescent sensor, that means we see some kind of chemical reaction in the paper. We can see fluorescent sensor, that means with the help of paper it gives fluorescent signal. Also electrochemical sensor, that means it gives some kind of voltage versus current graph with this paper-based sensor. Okay, so this is the your paper-based substrate or paper-based diagnostic system. So once we'll see a video here how they are made.
We have designed
See, this is self-powered paper-based diagnostic tools. So the top layer contains 4 electrochemical test areas. So you can see these are the screen printed electrodes. So they have made this screen printed electrode on top of a paper, simple paper. And 10 colorimetric tests. You can see this. On the bottom, there is a triboelectric generator that the user can tap to generate the peak voltage. Okay. So if when you touch a triboelectric generator, it will give a voltage, it will give a shock, and that is captured in the form of voltage. And that is transferred into this chip. Okay. These devices can be considered as portable laboratories. That a doctor can use in remote locations in order to assess or diagnose malnutrition problems. Okay. So this can detect kidney diseases, liver diseases, right? And even the quality of the drinkable water. We develop a machine vision algorithm that allows the user to take a picture of the device after doing a test and getting immediate results. So what they are doing, they are first they are testing it, they are taking the image of it, and now they are applying that image to the machine learning system, which based on the changes it will detect, the algorithm detect some results. See, just by identifying the colors on the devices, they are giving this result. Paper is a very good substrate because it is easily available. They are also biodegradable. They are cost effective, right, and easy to manufacture. You can see how they are disposing, right? So this is the advantage of paper-based diagnostic system. So we will do some this week's question first, then we'll come back here. So select the incorrect option with respect to paper-based diagnosis. Select the incorrect option with respect to paper-based diagnosis. So what are they? So we know that paper-based diagnosis is rapid. You can also comment on the chat also, right? The first question is very easy. That select the incorrect option with respect to paper-based diagnosis. It is rapid. We have just studied that paper-based diagnostic devices are rapid. They will give immediate results. They are inexpensive, right? And they use colorimetric assay. Like color change we see. The right pronunciation is colorimetric. Okay, so this is written as calorimetric. So this is the wrong answer. Okay. So this is wrong. So the incorrect option is the calorimetric assay. We'll come back to the next question because let's first discuss the transfer. Yes, this is the calorimetric assay. Okay. This is what are the three main paper-based diagnostic assays? What are the three main paper-based diagnostic assays? Tell me what are the three main. So, from the diagram itself, you can understand that first one is the dipstick assay. Dipstick assays means they are forming a strip kind of thing like this, and with different colors, and depending on the pH of the sample, they are detecting the amount of it. Okay, this is the dipstick method. Second one is the lateral flow assay, LFA. I'm writing in short form. This is LFA. So so in LFA there are four pads: sample pad, conjugation pad, the absorption sample pad, conjugation pad, absorption pad, and last one is the what is the last one? Yes, reaction pad. Reaction pad is the third one, and absorption pad is the last one, right? And the fourth one is the microfluidic paper-based devices, right? Third one is the microfluidic paper analytical devices or micropads. Okay. So here, this is the paper. They are fabricating with different patterns, and now they are sensing it with our samples, right? So this is the three type of paper-based diagnostic assays are there in the market or in the research. So you can see these videos of three different. Suppose this one is the dipstick assay. So this is a kind of strip available. This is the urine dipstick assay for detection of protein. Okay. So you can use it by your own in home. So dipstick for protein, the normal value is less than 15 mg per 24 hours. It's basically a semi-quantitative lesson. Urine protein expression is less than 150 milligrams. It can detect up to 15 milligrams per deciliter of algae. So now how they are detecting? So if you put one drop of urine on the strip, it will accordingly change the color, and that is how you can somehow quantify that in what range they are. Okay. So that is how dipstick assays work. Now comes the lateral flow assay. So you can see here. So first there is a sample pad, right? You can see. This is the one drop. This is the sample pad. Specific targets analyzed. So this is the sample pad where the drop is given, right? Now, what will they do? So this lateral flow assays are also used in time of COVID, in time of influenza virus, detect influenza virus, and in general the different kind of hormones in pregnancy test kit. Antibody is produced by the body in response to the presence of foreign substance such as bacteria, viruses, or allergens. An antigen is the specific protein that is recognized by the specific antibody. You can see this, right? So this is antibody is generated because of the foreign attacks, particle attacks or bacterial attacks, and these antigens are specific for binding to that particular antibody. So this could be a protein on the surface of the bacteria or virus or an allergen protein. The most common type of lateral flow assay are the sandwich immunoassays. Right? So you can see that your antibody is sandwiched between the two antigens. Right? In these assays, conjugate antibodies first bind an antigen. See, they are first binding to an antigen. This complex then travels towards the immobilized section, that is the by capillary action. And this antibody is again binded with the same kind of antigen in the absorption pad. This process of how this works in lateral flow device will be explained in this section. The main components of lateral flow assay. This is the sample pad what I am talking about. Here the sample is given. Our antibody is given here. Okay. This is here the sample is collected. Next is the conjugation pad. Here the sample meets the detection antibodies, okay, which is already stabilized on the conjugation pad. Third one is the nitrocellulose membrane, where this reaction happens and binds the target for color development. And fourth one is the wicking pad or absorption pad, where most things are absorbed and rest of the things are pulls out the remaining sample liquid. All these components are assembled together on an paper kind of thing. See, all these assemblies, because it's a paper-based diagnostic assay, right? So all these, every portion is attached adhesives by backing card and strips are commonly assembled into plastic housing box. So blood sample can be given, stool sample can be given, urine can be given. Also, they can be used in the environmental contaminant detection. Okay, so you can see the videos later. This is the basic. There are mostly 4 parts. Third one is the micropads. What are those? We'll see in a little video. See, this paper-based microfluidics is based on the capillary fluid. Okay, so if we put a drop of our sample on on this design of this paper, on this pattern, they will flow towards the other end because of the capillary pressure. Okay. They have gained popularity due to the increasing demand for affordable, portable, and user-friendly devices. A key element in these devices involves creating hydrophobic barrier for the movement of liquids. So, why hydrophobic barrier is needed? Because otherwise, if it is hydrophilic, because all our body samples are based on water, right? So that will flow in the other direction also. Suppose we want to make it flow to the direction of 4, but it is going out this side. It will spill, right? If these borders are made of hydrophobic barrier, then it will go. Then it will not cross the borders. It will go to its destination only. That is why hydrophobic barrier is very important. How to design? First, the design is done in the AutoCAD system. Will be to print the pattern from AutoCAD using a wax printer. Then we will infuse the wax into the paper, creating a hydrophobic barrier. See how they are creating a hydrophobic barrier by using wax. Yes. So, first the design is done, then they are printing it and they are making the border hydrophobic. Now they will put the samples and see the result. Okay. Print using the wax paper. So now this is printed. Heat wax. Right. Okay. Now see how this samples is given. Pipette is reagents to the detection zone. So these are the detection zones, 4 detection zones. Now the sample is given in the middle. These 4 are the detection zone and sample is given to the middle and it will be going to their destination because the background is hydrophobic. Can see. Detection zone color is changed little bit, right? You may not see properly because it is a light color changing, but the sign, some kind of yellowish thing you can see, I think. So that is your colorimetric. In other case, in other sample, it will be maybe it will give red color that will be more visible, right? So this is the colorimetric assay paper based. Now in details we will study the principle of lateral flow immunoassay. So you know, so this is the sample pad. The way I have said that in the sample pad, your our sample will be there, the antigen will be there, and in the conjugation pad, the antibody is coated with some kind of, with the help of nanomaterials, or we can say gold nanoparticles, right? They are already coated. And now they are moving to the nitrocellulose membrane. Here there are two lines, test line and the control line. If both the color changes in both the test lines and control line, then you will be thought of a positive patient. And if only the control line shows the color change, then you are a negative patient, right? So you can see now the antigen is conjugated with the antibody attached with nanoparticles, and now this is going to the test line, and in the test line it is sandwiched between the two antibodies, right? It is sandwiched between the two antibody, and suppose it is giving a color, and again it passes through the control line, and then also there is a monoclonal antibody and it shows some kind of color change, and now in the absorbent pad, extra flow is thrown away. Okay, so this is your lateral flow immunoassay. These four pads are very important. What are they? Sample pad, conjugation pad, reaction pad, where nitrocellulose membrane are there, right? And the bottom there are, and the end there are absorbent pad. Okay, so these are main parts of the lateral flow immunoassay. So how does pregnancy test it work? So this is also based on the lateral flow immunoassay. So we'll see this. So in the pregnancy test kit, what we are detecting? We are detecting a hormone, which is known as human chorionic gonadotropin hormone, HCG hormone in urine. So when someone is pregnant, so this HCG hormone will be available in the urine. Okay. So here, in the sample pad, that urine sample containing the HCG is given. So urine applied to the sample pad. If a woman is pregnant, urine contains HCG. Then comes the conjugation pad, right? Here already antibodies are there. So HCG binds to mobile antibodies. These antibodies also have an enzyme attached to them, right? Then it goes into the reaction pad. Here immobilized antibodies in the test zone bind to HCG and they are sandwiching the HCG hormone. The enzyme on the first antibody changes the test line color. Excess antibodies bind to immobilized antibodies in the control zone to show the test work correctly. Okay, and finally is the weakened zone. Here it is written much more clearly that first one is the hormone HCG is present in the pregnant woman's urine and will travel up to the test. Okay. In the second step, in the conjugation pad, HCG binds to placed monoclonal anti-HCG antibodies with enzyme that trigger the color change. Right? In the third step, already placed anti-HCG monoclonal antibodies bind to the already binded. So in the test line, they are between the hormone is becoming sandwiched between the two anti-HCG antibodies. Okay, and they are triggering a color change. Okay, and in the stage 4, which is already placed, monoclonal antibodies compatible to the anti-HCG antibodies bind to the anti-HCG and trigger a second color change. This is the control test that will cause a color change, not mattering the presence of HCG hormone. Okay, so that is the work of the control test, control line. Okay, so even if the HCG hormone is present or not, that line will change the color. Okay, so this is how the lateral flow assay or pregnancy test kit works. Next comes, how does a glucometer work? So glucometer is your handheld device. It is not paper based, but it is a POCD device. Okay, so how does it work? A glucometer works by using an enzyme on a test strip. So in the test strip already the glucose oxidase enzyme is present. Glucose oxidase. Glucose oxidase enzyme is present with a blood sample, and when we are giving a blood sample, it generates glucose oxidase, hydrolyzes the glucose that is present in our body, right, or in our blood sample, and it changes the reaction, and the enzymatic reaction happens there, and it generates an electric signal that the device measures. So it is kind of electrochemical based point of care device. Okay. So if you see the video, yes, I am coming to the question. This is a bit explaining a lot of things, so you can see on your own. I am showing a small video here. Okay, so this is a glucose monitoring device, right? So we use a blood. We first prick our fingers, and small amount of blood is coming out, and that we will keep on the paper strip of this glucometer device. So this is how blood is captured. The body is steadily releasing glucose into the bloodstream, which then moves into the interstitial fluid surrounding the body's cells before being absorbed by the cells for energy. Right? When taking a reading from a finger prick, you are measuring the concentration of glucose in the blood, which is referred to as a blood glucose reading. So how this is collecting your blood, this is showing here. Suppose this device is small sensor that measures glucose levels just beneath the skin. Okay, you can see I think you have understood the basic fundamental, how the glucometer works. So we can proceed to the next. So what is HCG? You know that HCG is the hormone, human chorionic gonadotropin hormone, right? So this is a heterodimeric glycoprotein with alpha and beta subunits. Next comes, what is NAT? You can tell me what is NAT? So NAT is the nucleic acid hybridization acid. So it uses two types of oligonucleotides, detector probe and capture probe. So they are in the NAT assay. One is the detector probe and another one is the capture probe, right? Detector and capture probes are both complementary with target nucleic acids, while the detector probe is used to combine with a tag to make the reaction visible or measurable. So in this image, you can see that this is some kind of nanoparticles, right? These nanoparticles are deposited on a graphene electrode, and now this S1 is our sample. It is given, and it is now MCH is given. It is also a part, or you can say some kind of external molecule that is reacted. Now, when the H2 is given, the reaction is happening, and you can see the LSPR enhancement here. Also, same way you can use the probe DNA and use a change in the electrochemical assay. That is also, this assay is based on the nucleic acid. That is why the name is nucleic acid hybridization assay. You can see here. Suppose we have isolated genomic DNA from the bacterial cells. We know that this DNA contains thousands of genes or DNA sequences encoding various proteins in the bacteria. Now we want to find out the presence of a specific DNA sequence in the bacterial genome. In other words, we are searching for a gene or DNA sequence of interest whose nucleotide composition is already known to us. But we want to find out whether this known sequence is present in the bacteria or not. And that for this reason, nucleic acid hybridization assay is used. So what are they doing? The red region in this DNA is representing the target sequence, and suppose the nucleotide sequence of this DNA portion is this in 5' to 3' direction, right? This is how the DNA is forming with the base pairs and so they will do the Southern blotting technique to find this. Okay. So here we need the nucleic acid hybridization, then the probes, and then the blotting. So denaturation and recession we have studied earlier class. So when the double-stranded DNA becomes a single-stranded DNA, separates DNA strand, this is the denaturation. When the heat is again given, it will renature, right? Now, if such denatured DNA strands are incubated under appropriate conditions, they will renature, right? To form a double-stranded molecule, and the basis of the spin iteration is the complementary base pairing between the two single-stranded DNA molecules. When two single-stranded nucleic acid molecules of complementary base sequence form a double-stranded hybrid, the process is known as nucleic acid hybridization. Okay, and now this thing is used as an acid. So you can see this is the nucleic acid hybridization 5,3 and 3,5. This is the complementary bases are attaching. Okay, again this call will end in 50 minutes, so I have to create a new one. First, let's do some question, then I will change the. So here we have new assignment, right? So first is done. Second one is the match the components involved in lateral flow assay with their correct function. So we have seen that the option is 2, right? So what is 2? Sample pad is the filters the impurities from sample and stores dried assay buffer, right? Then the conjugation pad. What does the conjugation pad do? This conjugation pad binding reaction between labels and the analyte parts. So you have seen in the lateral flow assay that first is the sample are given, then it is moving forward in the conjugation pad where it is binding with the antibodies. Then third one is the detection pad. It captures reagents are fixed and signal is developed. So it is reaction pad in the test line, and in the control line it is binding with the antigens, antibodies, and signaling a color change. And the last one is the absorbent pad where with the fluid through the membrane. So the right option is the 2. Next question. Which of the following routes should be avoided to prevent the destruction of the cargo content in the phagosomes from acidification and enzymolysis? So we'll come to the phagocytosis part, but if you have gone through the lectures already, if you could answer the question that in which route should be avoided to prevent the destruction of the cargo contained in the phagosomes from acidification and enzymolysis? Can you answer question 3? Yes, the question is answer is answer 3. So the answer is fusion of phagosome to lysosome. Question number 4. Hydrophilic nanoparticles have a higher affinity for the cell membrane than the hydrophobic ones. And statement 2 is PVP and dextran are hydrophilic polymers that form a cloud to suppress the interaction between the nanoparticles and lipid bilayers of cells. Choose the correct statement. Tell me. What is the answer of question 4? No, only statement A is not correct. Only statement B is correct. Because hydrophobic nanoparticles have a higher affinity for the cell membrane because cell membrane is negatively charged. So hydrophobic nanoparticles will have a kind of attraction towards the to the negative cell membrane, right? So that is why the statement A is wrong. Only the statement B is correct. Okay. From the question 5, we will do in the next call. So can you please join the call? I have can you please join the link I have shared in the chat? Because this call will end in 10 minutes. So please join that other call where I will be continuing with other questions. So please join the other call. Yes, so I think my screen is visible. So we'll continue from the question 5. The blood-brain barrier is composed of a network of endothelial cells which is not permeable to large molecular weight chemotherapeutic agents. And the statement 2 is the relative impermeability of blood-brain barrier is due to the tight junctions between capillary endothelial cells which are formed by cell adhesion molecules. Which one is true? Can anybody answer? BBB is composed of a network of endothelial cells which is not permeable to large molecular weight chemotherapeutic agents, and the relative impermeability of BBB is due to the tight junctions between capillary endothelial cells which are formed by cell adhesion molecules. Yes, both statements are correct. Next question. Which of the CNS disease requires large molecules drug therapy? Which of the CNS disease requires large molecule drug therapy? Yes, the answer is Parkinson's disease. Next is the ISO and BSI agencies that sets regulations to nanotechnology stands for. So I think this is very easy. So it is International Standards Organization and British Standards Institute. So it is also option A. Next, acridine orange is a fluorescent dye, fluorescent DNA intercalating dye, permeable to all cell nuclei, enters only the membrane with compromised nucleus, emits red-orange color fluorescence. Which one is true? Question 8. Acridine orange is a fluorescent dye and permeable to all cell nuclei, enters only the membrane compromised nucleus and emits red-orange color fluorescence. Select the correct option. Correct option. The answer is 1, 2. So both first statement and second statement are correct. So acridine orange, even if it it states like acridine orange, but it shows a fluorescent DNA intercalating dye. It shows fluorescent color and it is permeable to all cell nuclei. So the answer is 1 and 2. Next. Trypan blue dye used to stain the live cells only, stain the dead cells only, helps to distinguish between the apoptotic cell and necrotic cell, measure the cell viability, and also known as dye exclusion method. So which options are correct about the trypan blue? Answer 2. That is 2, 3, 4. Answer 2 is correct. It is the stain date cell. It stains date cell. And also the 4 is correct. Measure the cell viability and also known as dye exclusion method. But because in case of apoptotic cell and necrotic cell, cell death happens in both of the cases, right? So when the trypan blue is able to detect the dead, able to stain the dead cells only. So the cell is that from the apoptotic cell death or necrotic cell death, you don't know. So I think option 3 is not correct here. I might be wrong, but I am giving here 2 and 4 option, but you just check it, okay? For to me, that option 2 and option 4 are correct because in case of apoptosis and necrosis, in both the cases, cells eventually dies, right? So you cannot distinguish that from which it is dead. So it will be not distinguished by the staining color, I think so. You may cross check also, okay. The last question is, select the in vitro methods used for measuring the antibacterial properties of nanomaterials. Select the in vitro methods used for measuring the antibacterial properties of nanomaterials. Colony counting method, optical density measurements, distribution method, or all of the above. Yes, this is all of the above. We'll do some old assignment question also. So first question is, most of the point of care test systems are based on the principle of easy to use membrane based test strips. And statement 2 is, point of care systems can be handled only by medical professionals with advanced clinical knowledge. Which among the above statements are correct, do you think? So first one, it says that it is based on the test strips, use of membrane based test strip, and second it is saying that it is only can be handled by medical professionals. Which one is true? So the right answer will be only one, right? The statement 1 is correct. Because you know that the point of care testing is developed because anyone can use that, right? So the statement 2 is completely wrong. So the answer is 1 only. Next, the pregnancy test assay detects the presence of the hormone in the sample of women. Like which hormone it uses and what type of sample, and this hormone is available in where, like in blood or urine. So this is a very easy question. You should be able to answer. Can you tell? Question number 2. The pregnancy test assay detects the presence of the hormone name, and what type of sample is that? The answer is human chorionic gonadotropin HCG, and it is available in urine sample. Okay. Next question. To facilitate electrostatic interaction of a drug with DNA molecule, the drug should be what? To facilitate electrostatic interaction of a drug with a DNA molecule, the drug should be what? Please answer. Electrostatic interaction means the interaction of the positive and negative charge. So it will be positively charged. Okay. So that to facilitate electrostatic interaction of a drug with a DNA molecule, the drug should be positively charged. Next, the term ADME in pharmacology refers to what? The term ADME in pharmacology refers to what? It is the absorption, distribution, metabolism, and excretion. Okay, so it is 4. Absorption, distribution, metabolism, and excretion. Next, phagocytosis is a type of what? Phagocytosis is a type of what? Tell me. Phagocytosis is a type of what among these 4 options? No, it is endocytosis. So phagocytosis is about, we'll discuss phagocytosis, but right now if I tell you that phagocytosis is like cell gobbles up a particle. Suppose this is a particle and this is a cell membrane. So what happens? The cell eats that particle like this. Okay, so this is the gobbling of the particle of the cell. So this is endocytosis. That means the nanoparticles or something, some molecules also is gobbled up. That is, it is coming to the cell or cell membrane. When the molecules is going out from the cell membrane, if the direction of this phenomena is opposite, that is exocytosis. Okay. So the right answer is endocytosis. Next question: hypertonic sucrose and chlorpromazine are inhibitors of endocytosis. This is just information. You can, when we will go through the lectures, you will find these are inhibitors of endocytosis. Okay, we'll come back to this question later. Let's study something else. So where were we? So we have studied this nucleic acid hybridization assay, right? So these are some of examples of these POCT devices. We'll discuss it at the end. First, complete the chapters like what are target drug interactions? What are target drug interactions? So, target drug interactions are based on pharmacokinetic interactions, pharmacokinetic and pharmacodynamic interaction. Okay, what does this mean? So, pharmacokinetics is defined as the quantitative analysis of the process of drug absorption, distribution, and elimination that determine the time course of drug action in response to an administered drug dose. Okay, and this is pharmacodynamics deals with the mechanism of drug action. Basically, in the simple term, pharmacokinetic is the body effect due to drug intake, and pharmacodynamic is the study of drug effect on body. Okay. Okay, in the simple term, we'll remember like this. Pharmacokinetic means the body, what body does to drug. Okay. So this one is the what body does to drugs, and this one pharmacodynamics is the what drug does to the body. This vice versa study is called the pharmacokinetics and pharmacodynamics, and these interactions are based on, like pharmacokinetic interactions are based on absorption, distribution, biotransformation, and excretion, right? So these are based on absorption, distribution, biotransformation, and excretion. And pharmacodynamics are dependent upon receptor interaction. That means how drug will enter. That is what I had said, right? That what drug does to the body is the pharmacodynamics. So first, receptor interaction. Second one is the receptor sensitivity. And third one is the drug transportation. So these are the interactions of pharmacokinetic and pharmacodynamics, which is based on our target drug interactions. Okay. So if you see the difference here, so pharmacokinetics focuses on how the body affects the drug. Okay, that is what I told. What body does to a drug. How the body affects a drug involving absorption, distribution, metabolism, and excretion. And pharmacodynamics deals with how a drug affects the body, including its mechanism of action and the resulting physiological effects. Second point is examines the drug's journey through the body from absorption into the bloodstream to its elimination or excretion. And This represents, pharmacodynamics represents the number of atoms of a particular element within a molecule. Pharmacokinetics involves measurements like absorption rate, bioavailability, half-life, and clearance. And pharmacodynamics involves parameters such as efficacy, potency, and the dose response relationship. Okay, I think it is clear. Next, what are entrapment and encapsulation of drug? What are entrapment and encapsulation of drug? So, entrapment means suspension of drug molecules within a polymer matrix, right? Suppose this is a polymer matrix and drugs are encapsulated like this. Okay, this is the drug entrapment. And next one is the encapsulation. So it is the coating of or surrounding drug molecules with a polymer coating, with a solid polymer coating or shell. So here these small particles are our drug, and this is the polymer matrix. And in encapsulation, the core is made of drug. The core is drug that is encapsulated by a solid polymer shell. Okay. So this is the difference between entrapment and encapsulation of drug. Next, what is nanomedicine? Okay. So nanomedicine applies nanotechnology to develop tools for diagnosing, preventing, and treating diseases at a molecular level using nanomaterials like nanoparticles to target specific cells, deliver drugs more effectively, and improve medical imaging. The tiny size of these materials allows them to interact with biological processes at the cellular and even DNA level, offering personalized medicine solutions by enhancing therapeutic efficacy and reducing side effects. So this is the nanomedicine basically is based on the nanotechnology dependent diagnosing and its cure. Okay, where nanoparticles or any kind of nanomaterials are the most important parameters. Okay, and these can be used to kill target cells. In the last week's class, we have seen that how these theranostic materials or theranostic process is killing the cancer cells particularly, right, with the help of nanomaterials. So that is also one kind of nanomedicine. Also, we have seen how the drug delivery is possible with the help of nanoparticles. This is the part of nanomedicine. Then to observe the effect of a particular reaction with the medical imaging with quantum dots, kind of small using small nanomaterials like quantum dots, the bioimaging is possible with their fluorescent activities, fluorescent characteristics. So all these come under the nanomedicine applications. Okay. So you can see it is a diagnostics, in vitro, in vivo, medical imaging, nanotherapeutics, vaccines, and regenerative medicine. Okay. So we'll see one small video. Other video you can see later. So nanomedicine means it is based on completely nano technology. Okay. So nano means dwarf. Dwarf means small thing. And nanotechnology, technology to do with small things. Okay. Nano also means a billion. For example, nanometer is a billionth of a meter. To make things more visual, a marble is a billionth in size when the size is of Earth, right? Hence, nanotechnology is science, engineering, and technology conducted at the nanoscale. The ideas and concepts of nanotechnology started. This is the history. So how you can see that how this nanoparticle and nanotechnology is used in everyday life in our suns, in our any cosmetic product, in sunglasses, in smart windows, everywhere. Nanoparticles use of nanotechnology is getting high and high. So nanomedicine or nanotechnology enabled medicine is one part of that. So nanomedicine is used in diagnostics, treatment, and they are more advantageous compared to the traditional mechanics. Traditional can really be only detect when a visible change the tissues has occurred, at which point thousands of cells have already multiplied and may be metastasized. Additionally, current imaging technologies can really only identify the presence of cancer, but identifying the characteristics of disease, including the nature of the tumor, requires biopsy. Right? These are the two shortcomings of the nanotechnology. Has potential to address these two shortcomings of the conventional technology. First one, it detects change after it has worked and does not identify characteristics of disease. Antibodies which binds to receptors commonly found on cancer cells can be coated onto nanoparticles which can be detected using diagnostic imaging technologies such as MRI and CT scan. Furthermore, this concept can be applied to various molecular, which means specific targets and types of cancer could be identified using nanotechnology. Right? Aside from its applications in diagnosing diseases, there are many applications. Which are they? Cancer treatment, drug delivery systems, then chemotherapy, which involves the use of medications to kill cancers. However, there are several issues with chemotherapy. Last week we have discussed that how chemotherapy kills the healthy cells also, right? Nanoparticles have the potential to more effectively target only the cancer cells and therefore minimize the harms. Furthermore, the specific size of nanoparticles, which range from 10 to 100 nanometers, allow them to be large enough while small enough to avoid immune detection. In other words, the drugs which the nanoparticles are delivering are more likely to reach their target cell, the cancer, and spare the healthy cells in the body. Finally, one more example is the includes the recent COVID-19 mRNA vaccines by Pfizer and Moderna. Right? What they did? Nanoparticles. Lipid nanoparticles in specific are used in both this module, vaccines to counteract two of the main viruses that are mRNA. The first challenge posed by utilizing mRNA is that there are many RNA enzymes, right, which could destroy the mRNA. To counteract these, both the Pfizer and Moderna vaccines encapsulates the mRNA with lipid nanoparticles as a barrier to protect the mRNA. Additionally, because our cells are surrounded by membranes and charged, it is difficult for the negatively charged mRNA to cross the barrier. This is where nanoparticles come into effect. As they facilitate transport of mRNA into the target cells, these are just a few examples of application. So what are these, in the COVID vaccine, mRNA can be killed by the enzymes, right? So these lipid nanoparticles are protecting this mRNA from the external attack. So this is one kind of nanomedicine, huge application of nanomedicine. So what are the new research developments? Medically assisted reproduction. Okay, so you can see it later also. Another video you can see. I'm proceeding to the next question. Next question is what are the major entries of nanoparticles in the body? What are the major entries of nanoparticles in the body? But how nanoparticles can enter into your body? How nanoparticles can enter through your body? So there are dermal routes. That means with the skin. Dermal means with our skin, with the pores of our skin. Nanoparticles are such small particles, they can infuse through our skin or dermal. Okay. Then the ocular. Ocular means through eyes. Okay. Then the respiration or respiratory system. That means when we are inhaling, nanoparticles also can come. And through the gastrointestinal system. Okay, these are the 4 ways nanoparticles can enter into our body. Next, nanoparticles internalization pathways in mammalian cells. So now nanoparticles are coming in our blood, but from blood, how they are going to our particular cells where it needs to be acted, right? So how that is reaching in the particular cell, this internalization process in mammalian cells, there are 5 or 6 processes. Okay. So what are these 5 or 6 processes? So this is called the endocytosis based internalization. Internalization of the particle means endocytosis. That means particles are coming into the cell. And exocytosis means the molecules or particles are going out of the cell. Okay, endo and exo. So endocytosis based is divided in 3 or 2 divisions, mostly. So what are they? I am showing in another. So this process of cellular uptake of nanoparticle is called the endocytosis. Then this is divided in 2. First one is the phagocytosis. Second one is the pinocytosis. Okay. So phagocytosis means the cell eating and pinocytosis means the cell drink. This is cell eating and this one is cell drink. Now this pinocytosis is again divided into four subsections. First one is the macropinocytosis. Then comes the clustering dependent pinocytosis. Then comes the caveoli dependent pinocytosis. And last comes the clustering and caveolin independent pinocytosis. Okay, these are the most main types of endocytosis or cellular uptake process. So here you can see this is the endocytosis based. So it is first one is the clustering dependent. So you can see this is the molecule. We'll discuss each of the endocytosis in detail, but first I am giving the overview. So first one is there, you can see there, this is a particle. Clustering membrane is forming around the particle. Now this is squeezing this cell membrane and that is pinching of the droplet. Okay. Now this clustering layer will be disassembled and this our target nanoparticle will go into the endosome, right? Next one is the caveolin dependent. Similarly, here also caveolin layer will form here. Same way, the caveolin coated vesicle will be formed, and now then the caveosomes will happen here, and they will release the particle. Clustering caveolin independent particles are independent pinocytosis are this different kind of fast endothelin mediated endocytosis. Okay, how can you can see there? This is CLIC and GEC. This is the other molecule associated endocytosis. Next is the macropinocytosis, means the cell is engulfing the nanoparticle and its cellular matrix together, and this forms this kind of structure. And phagocytosis means this is just eating the cell from both sides. If from the diagram itself you can understand it is just eating the cell, right? And direct cellular entry is also there, that direct translocations of molecule from one side of the cell to the inside of the cell. This is a lipid fusion, then the electroporation, this and other one is the microinjection. Okay. So now we see this one by one. So this is the typical range of the particles for different type of endocytosis. So phagocytosis generally depends on the particle size. So this bigger particle will be captured by the phagocytosis. Okay. And when the particle size is less than greater than 1 micrometer, then the macro pinocytosis will happen. like this. One cell will be taking up from one side. Then the for clathrin mediated endocytosis, there is 120 nanometer particle size. For caveolin, again smaller, this is the 6 nanometer particle size, and clathrin caveolin independent endocytosis, it is for 90 nanometer particle and cellular matrix. Okay, we will see on video. This is talking about overall endocytosis. So you can see here from the start. The cell needs to take in substance, right, for many functions from outside to inside. So very small particles can penetrate the lipid bilayer itself or pass. This is the direct cell entry, what I have shown, right? However, very large particles cannot cross the membrane. Okay. For that is why, so they are taken inside by the cell by a special process called endocytosis. Basically, in endocytosis, the cell membrane wraps around the particle just like that. You can see vesicle containing the particle inside the cell. Okay, and based on how it is formed, you can understand that the fluid inside this vesicle, the extracellular fluid from the outside and the wall of the vesicle is a detached proton of the portion of the cell membrane, right? So these are the basics. So based on this cell membrane and based on this external layer, what is forming around this nanoparticle, this endocytosis is divided into phagocytosis and pinocytosis, and then again this pinocytosis is divided into subgroups. So the main is pinocytosis and phagocytosis, right? First, let's talk about the pinocytosis, which have many subgroups. Means that drinking by the cell, right? I have told this earlier. It occurs to take in macromolecules. The cell expresses receptors for such molecules on the membrane, right? Such receptors are generally concentrated in a small pit on the membrane. It is called the coated pit. Beneath this pit, on the inner side of the cell, there is a lattice work or fibrillar proteins called clathrin and other proteins like contractile filaments, actin and myosin. Okay. So these are the fibril proteins. Okay. Now what will happen? All these proteins drive the movement of the cell membrane during endocytosis. What happens is when the particle attaches to the receptor on the outer side of the wall, these proteins pull the membrane inside the cell, thus deepening the pit. See how these proteins, this clathrin protein and actin and myosin, how all these things are helping these molecules to get inside the cell. See how this pit is depending. Then they close the mouth of this pit, forming a vesicle inside the cell, right? These proteins dissociate from the vesicle and go back to the membrane for next round of endocytosis. This entire process requires internal energy from ATP. The vesicle formed by pinocytosis is called pinocytotic vesicle. Such vesicles are very small. Pinocytosis occurs in almost all the cells in the body. So this was about the pinocytosis. So different types of pinocytosis, they will show it later. What is phagocytosis? Phagocytosis is the cell eating, right? It occurs for the intake of large particles such as bacteria or whole cells, etc. So see, generally bacteria size is around 2 micrometer to 10 micrometer. This 2 micrometer is still big or large particle. This 2 micrometer bacteria is also considered as large inside our body. So you can imagine that how our body works. Only some cells are capable of doing phagocytosis, like tissue macrophages and some white blood cells. The process of phagocytosis is similar to pinocytosis. First, some ligand on the particle to be engulfed binds with the receptor on the cell. Okay. This initiates the process of phagocytosis. In this, the membrane around the points of attachment evaginates outwards to surround. So, in the pinocytosis, what was happening? The cell membrane was deepening, right? But in case of phagocytosis, what is happening? The cell membrane is engulfing or evaginates outwards, right? This is the two opposite phenomena. The thing is happening same. The process is same, but it is happening in the initiation of the process is a little bit different. Initially, in the pinocytosis, it is deepening the cell membrane, and in case of this phagocytosis, cell membrane is engulfing by evaginating itself to surround the entire particle, right? At the tip, the membrane march creating a vesicle inside the cell. It is called a phagocytic cell. So this is how phagocytosis happens. Now the cell has organelles called lysosomes containing various hydrolase enzymes, right? The vesicle formed by pinocytosis or phagocytosis march with this lysosome. This empties the enzymes in lysosomes in the vesicle. Okay. Now it's called the digestive vesicle. And as per the name, it starts digesting the proteins, carbohydrates, lipids, and others. Okay. Inside the vesicle. Small molecules like amino acids, glucose, phosphate, etc. formed during these digestions, cross the membrane and enter the cytoplasm. But indigestible things are left inside the vesicle. They are excreted out of the cell by exocytosis. Right? So this is all about the ingestion of by the cell. You can see how this is done. How this beautifully this endocytosis process is explained. Right? I hope now the endocytosis and exocytosis is little bit clear. So phagocytosis is what we have just seen in the video. See, this is the microbe or other particle. This is the plasma membrane. This is the pseudopod. This is the cytoplasm inside the cell. So in the first, chemotaxis and adherence of microbe to the phagocyte, ingestion of the microbe by phagocyte, right? Then the formation of phagosomes, fusion of the phagosome with the lysosome to form a phagolysosome. Then in the step 5, digestion of ingested microbe by enzyme and formation of the residual body by containing indigestible material and discharge of this material by the exocytosis process. Right, so What is written? Phagocytosis is a cellular process where a phagocyte uses its plasma membrane to engulf large particles such as bacteria, cellular debris, or dead cells, bringing them into the cell to form a phagosome. Once inside, the phagosome fuses with lysosomes to form a phagolysosome, where the particle is degraded and destroyed. This process is a type of endocytosis crucial for the immune system to clear pathogens and for tissue homeostasis. So there is a video you can see later. I am not showing it now. Next is the pinocytosis. So you already know now, I think it is clear what is pinocytosis. These are the solutes bind to the cell membrane. The cell is deepening. Cell membrane starts to fold inwards. Then cell membrane continues to fold. Both ends of the fold meet, producing a pinosome. Right? So pinocytosis is the process by which a cell takes in fluids and dissolves substances from its surroundings by forming small fluid-filled vesicles. The cell membrane folds inward, creating a pocket that engulfs the fluid and then pinches off it to form a small vesicle, trapping the fluid and its contents inside the cell. Okay. So here also one video is given, you can see later. Now comes the different types of pinocytosis. This is CME, this is the clastrin, this is the clastrin mediated endocytosis. So here you can see, this is the, here the receptor is coming, here the particle is coming, here are the, these are the receptors, cell receptors. They are forming a clastrin coated peak. This clastrin is the fibril protein and they are forming here, and now this clastrin coated vesicle is formed from this, and now these proteins are taken out, and this is now has become a naked vesicle, and this is going for early embryo. So, clathrin-dependent endocytosis is a cellular process where the cell membrane invaginates to form a pit, then pinches off to create a clathrin-coated vesicle that internalizes extracellular matrix or molecules like nutrients, hormones, and receptors. This receptor-mediated endocytosis pathway relies on the protein clathrin, which self-assembles into a lattice-like coat on the inward bending membrane. Okay. Adaptase proteins link specific cargo to the clathrin coat, while dynamin facilitates the final scission of to form the clathrin-coated vesicle, which then travels within the cell. I am showing this video because next comes the caveolin mediated endocytosis, so you will understand the difference between these two. Right? So it is saying that some specific organelles inside the cell need specialized vesicles which are just like the trucks which would leave the warehouse to deliver the particles, parcels in specific locations in the city. So in this vesicles, clathrin coated vesicles. Okay. Clathrin coated vesicles. So as the name suggests, it is the clathrin coated. So it is coated by clathrin, and clathrin is produced by a complicated lattice of triskelion molecules. The triskelion molecules, as depicted here, has a heavy chain of 180 kilodalton and light chain of 35 kilodalton. and these crystalline residues attaches to each other at their head domain and form a complicated clustering lattice all around the vesicle. So this is the ensured the vesicle safety while it is transporting. So here it is coming through the cell membrane. The ligand inside several adapter proteins such as beta-arrestin would invite other adapters like a protein is very important because adapter protein 2 serves as a platform where these prion molecules can associate and build up in format of a vesicle. So you can see here how this membrane is forming a clustering coated vesicle. Right? Next comes the caveoli dependent endocytosis. So in the case of in this case, a clathrin, a caveoli membrane is formed around this vesicle like this. Okay. So what does it say? So caveoli dependent endocytosis is a clathrin independent mechanism for internalizing molecules into cells through invaginations of the plasma membrane called caveoli, which are enriched in cholesterol, sphingolipids, and protein caveoli. This process mediates the uptake of various substances, including some viruses. So, caveoli-dependent endocytosis helps in uptaking small particles, right? That is why viruses are coming here. And certain receptors and plays role in cell signaling, migration, and polarity. Key steps involve the formation of cholesterol-rich invaginations stabilized by caveolin 1 and internalization via a dynamine-dependent or independent process into vesicles that can form caveosomes or fuse with early endosomes. So this is the caveolin-dependent endocytosis. This is the accelerometric matrix, this is the cytosol, and how it is coming will see. See how this membrane is formed. Lipid drugs associated with certain types of receptors occurs with the help of proteins called as caveolin. In the earlier case, this is the clathrin protein, and in now this is the caveolin protein. Okay. Then causes release of this endocytic vesicles from the plasma membrane to the cytosol. This is the endocytic vessel. Right? Now this is becoming the caveosomes. This endocytic vessel diffuses with the caveosomes due to this fusion of the molecules present in the endocytic vesicles, and these are internalized into the caveosome. Internalized molecules are then sorted, and caveosome containing sorted internalized molecules then moves towards early endosome and fuses with the early endosome. The early endosome diffusion delivers the sorted internalized molecules from caveosome to early endosome are then prepared for recycling to the plasma membrane surface. In another way, in eukaryotic cells, first of all, invagination of patches of plasma membrane containing lipid wraps associated with the certain types of receptors. So you can see it later. So basically, the principle is very easy to understand. Next, from the macropinocytosis, it is the where cell non-selectively engulfs large volumes of extracellular fluid and solutes into large internal vesicles called macropinosomes, essentially a form of cellular drinking. This actin-dependent mechanism is vital in various physiological processes, including feeding in amoeba, nutrient scavenging for cancer cells, and immune surveillance by antigen-presenting cells. It also serves as an entry point for some pathogens and can be exploited for targeted cancer therapies by delivering drugs or blocking nutrient uptake. You can see the video later. So this is this. You can see this is the macropinocytosis, right? The cell is going out and collecting this particle along with the cellular matrix and forming the macropinosome. Then this is the clathrin-mediated endocytosis. These are clathrin-coated vesicles. And finally, they are mixing with the lysosome. This is the caveoli-mediated endosome. Okay, this is the caveolar vesicle. And finally, they are forming this Golgi and the endoplasmic reticulum. Okay. This clathrin and caveoli independent endocytosis is refers to the cellular process where cargo is internalized without using clathrin-coated or pits or caveoli-coated pits, the two most common endocytic mechanisms. Instead, these pathways often involve lipid wraps, depend on specific molecular machinery. So, these caveoli protein and clathrin protein are not being used here. Rather, they are using some kind of lipids, some kind of specific molecular machinery like the GTPases and the cholesterol and can be have high capacity or leverage by pathogens for entry into cells. Okay. This is the clathrin and caveoli independent endocytosis. Next, we will go to the apoptosis, but before that, we can study a little bit questions which are left to do. So, then the lowest concentration of nanoparticles that inhibits the growth of bacteria is called the lowest concentration of nanoparticles that inhibits the growth of bacteria is called what? Can you answer? The lowest concentration of the nanoparticles that inhibits the growth of bacteria is called the minimum inhibitory concentration. The zone of incubation is a term associated with what? The zone of incubation is a term associated with the zone of incubation is a term associated with yes, this disk diffusion. Okay. Serial dilution or disk diffusion? I think it's disk diffusion. Next is the gold nanoparticles are highly inactive and gold nanoparticle is an inert material. Which one is true? Which one is true? Gold nanoparticles are highly inactive or gold nanoparticle is an inert material? Which one is true? Both A and R true, or R is the correct explanation of A? Both A and R true, but R is not the correct explanation of A. A is true, but R is false, or A is false, but R is true. Which one? So the answer is A is false, but R is true, because you know that gold nanoparticles are highly active, and these are mostly the particles used in this all type of nanotechnology, nanomedicine. And gold is an inert material. That it means it doesn't react with any other thing. Last question is that consider the following models of testing toxicity of nanoparticles: cell lines, bacteria, zebrafish, and mouse model. Which among the above are the examples of in vivo testing? So for case of in vivo testing, zebrafish model and mouse models are used. Okay, so the answer is C and 4. Next. The sandwich ELISA is typically used for what? The sandwich ELISA is typically used for what? So if you have gone through the lectures, you will be able to answer that. The sandwich ELISA is typically used for detecting multiple antigen epitopes. So this is option B, detecting multiple antigen epitopes. Next comes, consider the following parts of the lateral flow assay strip. So first one is the sample pad, then is the conjugation pad, then detection pad, then absorbent pad, right? So 2, 1, 4, 3. Okay, so this is the 2, 1, 4, 3. Next, controlled drug release follows first order kinetics, second order kinetics, zero order kinetics, pseudo first order kinetics. So controlled drug release means it will not depend on any time factor. So it is independent of the time factor. So it is a zero order kinetics. Next question, human on a chip platforms employed for in vitro assessment of ADME properties and drug design can be utilized to study the effect of drug on the particular organ, know how the drug can be excreted, physiologically based pharmacokinetic models in which organs are represented by an actual compartment, and number 4, predict the potential toxic side effects with higher accuracy. Choose the correct option. Tell me. So, human on a chip model is used for all the 4 things. So, it is the option 4. Next, choose the correct statements regarding the macropinocytosis pathway. What is that? Present in nearly all cells except those of brain microvessel endothelium. Activation of receptor tyrosine kinases occurs due to external stimuli. Opsonized nanoparticles attach to the cell surface and trigger the formation of cup-shaped membrane extensions. And 4, specific lipid composition including cholesterol is essential for its function. So, in case of 5, for macropinocytosis, it is present in all the cells except the brain vessel, and number 2, activation of receptor tyrosine kinase occurs. So, it is 1 and 2 only. Next is the Inhibition of clathrin-independent endocytosis can be achieved using hypertonic sucrose solution with a concentration ranging from 0.4 to 0.5 molar. Caveoli-dependent endocytosis can be inhibited by employing cholesterol oxidase. Clathrin-dependent endocytosis is blocked by the action of amiloride, and choose the correct option. So, clathrin-independent endocytosis can be inhibited by using the sucrose solution. 1 is correct and 3 is correct. The clathrin-dependent endocytosis is blocked by the action of amiloride. Okay. Next, which of the following fluorescent dye stains nucleus? So tell me, which of the following dye stains nucleus? It is the first one. Okay, because rhodamine B dyes the cell membrane, trypan blue dyes the stains the dead cells only. So for nucleus, it is the OCHST 3334. Okay. Next question: which of the following statement accurately describes the acridine orange? So we have done this today. So it is the acridine orange is a fluorescent dye that intercalates with DNA, and acridine orange can permeate all cell nuclei. So it is only the 1 and 2. Next question. Zebrafish can be a potential human model system because of the following reasons: transparent embryos, functional homologue of -70% of human diseases, forward and genetic model system, short maturation time of the embryo. So this is the all 4. The correct option is 1, 2, 3, 4. And the last question is, quantum dots toxicity study in liver cells was lowered when measured using 2D scaffold technique. And statement 2 is, substituting 2D in vitro assays for 3D ones increases the accuracy of the test. Tell me which one is the correct option. So it is written that quantum dot toxicity study in liver cells was lowered when it is used 3D scaffold technique. Okay. So statement 1 is wrong. And 3D one increases the accuracy of the test. So only the statement 2 is true. Okay. Okay. We are leaving with some extra questions. So what is that apoptosis? So you know that apoptosis, you can see the video later by your own. I am not going through the video. I am just explaining what is apoptosis. So apoptosis is a programmed cell death, right? An organized energy dependent process where cells systematically dismantle themselves in a controlled manner to remove unneeded, damaged, or abnormal cells without causing harm to surrounding tissues. Okay, so apoptosis is one type of cell death, but it is programmed, but it is planned cell death. Okay, it is not unwanted. It is required for the body. It is essential for proper development and maintaining the balance of cells in the body, contrasting with necrosis, which is uncontrolled cell death from injury that causes inflammation. There are 4 stages of apoptosis. First one is the initiation or induction, when a signal triggers the process. The early or mid stage involving caspase activation, cell shrinkage, and chromatin condensation. Third one is the late stage, characterized by membrane bleeding, formation of apoptotic body, that means small another cells, and DNA fragmentation. And the last one is the clearance or phagocytosis, where macrophages engulf the apoptotic body. Okay, so from this image you can see that first there is a normal cell, then the shrinkage happens, then the membrane bleeding happens, right? And then it breaks into small bodies which are called apoptotic bodies. and these bodies are engulfed by macrophages by phagocytosis process. Okay, this is the apoptosis. What is necrosis? Necrosis is the complete death of the cell or unnatural death of the cell after injury. So you can see the comparison here. Which is the programmed death? This is the cell death after injury. So here you can see nuclear is fragmented, but here it is swelling. The cell is swelling. It is shrinking and it is forming apoptotic bodies. But here some cell is swelling, but cell leakage is happening. Means the cytoplasm inside the cells are leaking out of the cell. Now this will completely destroy the cells after some time, and it will lead to some kind of inflammatory response. And here these debris are swallowed by these macrophages. So no inflammatory reaction. So this is the basic difference between the apoptosis and necrosis. You can go through the video here. So I am just reading it. Necrosis is an irreversible death. So after the apoptosis, you can recover the cell, right? Because you are engulfing this. But in necrosis, it is the irreversible cell death and tissues in a living organism, typically caused by lack of blood supply, infection, injury, or toxins, rather than programmed cell death. It is an uncontrolled process that leads to cell swelling and rupture, releasing its contents into surrounding tissues and often triggering an inflammatory response. Symptoms can include pain, swelling, and visible skin changes. And affected tissue may be firm or leathery or moist and stringy, depending on the types of necrosis. Okay. So this is the difference between apoptosis and necrosis. Last, I will talk about these facts, which is the flow-assisted cytometry, cell cytometry. This is the flow-activated or assisted flow-activated cell sorting. Also, it is called flow cytometry. Okay, what is happening here? So, all kind of cells like dead cells and live cells are passing through this channel, and where there is a sheath of fluid. Okay, and now this laser is uh, incident on the cells, and by the scattering of the cells, this is detected. Okay, so there are two type of detectors. One detected the size of healthy or dead cell. One detected the fluorescence response of the cells. So by the after the laser incident, this scatters, the cell scatters, and it detects through this, and at the outside there is the charged molecules are there. Okay, they are, so at the outside there are charged molecules or electrodes are there, or electromagnets are there. What is happening now? They are separating this cells, this positive, this dead cells and healthy cells in the different shells. And this is the instrument of the flow cytometry. One, just we will see this video last. So this is the instrument of the flow cytometry or FACS. Okay. You see, according to the size or cellular complexity, it is separated. It could be used to analyze cell cycle, cell viability, and many other aspects. So, what is the mechanism? See, this is the fluidic system. This is the heart of the system. The group we will talk a lot about the fluidic system because the principles which are used in this fluidic system is very important. Then a detector system. So a light is required to illuminate the samples. It's not similar like a microscope. So the laser modules are important component. Now the lasers that are emitted has to be detected by something like light so that they are multiple detection system. Mostly photomultiplier tubes are used for the detection. Okay. This is the main fluidic concept is the hydrodynamic focusing. This is the focus. So hydro. Let's talk about that. What is that hydrodynamics? There is a fluid which is a light blue fluid inside, and there are like multiple different cells inside that fluid suspended the fluid. Now there would be another fluid surrounding that fluid stream. So this is called the sheath fluid. And if the sheath fluid is flowing at a very high rate, it would focus all the substances inside the pore fluid into a streamline motion. Right? So in hydrodynamic focusing, the sheath fluid and the pore fluid system plays a vital role, and it ensures that at the end all the particles are traveling at the same axis with the approximate same velocity. So this is the core concept of a flow cytometry. Okay, you can see the details here. Next, how are they sorting? The same velocity, and now a laser is a pose intersection point, but make sure the laser should not directly hit the detector. Otherwise, the detector would go blind. Yes. So it is written in very detail. You can study it later. So what is happening? And just so this is how the laser is falling upon the cell, and it is getting detected. And in the what happens? If the cell is small, the forward scatter would be small, right? But if the forward scatter light can be also bent into higher angles, which would be detected by side scatter detectors. Now, light would be scattered in higher angles when the cellular complexity is more, when the light would really get a lot of obstruction in while passing through the sample. So a complex sample would scatter light in more different directions. So the side scatter would be more for a very complex sample. Right? Side scatter means depending on the complexity and granularity. You can see it later. I think you have understood the basic concept. Last thing I need to tell this have shown some examples. I will be sharing this PPT. You can take it later also. I am giving the examples of some kind of POCT devices developed in our lab for detecting. This is for detecting the lipase. So you know that lipase hydrolyzes the olive oil and form the monohydrate fatty acid, right? So what happens initially when olive oil is given it in water, it is showing a blue color. Now, when this it is showing a green color, right? Now, when this lipase is given, it breaks the olive oil chain, it breaks the fatty acid, and fatty acid change and taken out the monohydrate dihydrate fatty acid, right? And now the color changes as the fatty acid is generated in the solution. The color changes little bit to the yellow side because we are giving an indicator like bromothymol blue. And that color change is detected by this LED LDR system. LED LDR means we are taking a particular LED light, which is the highly active at particular wavelength. This solution is highly active for this red LED. And now when this LDR means the detector, LDR is one kind of detector. So uh, this is based on the resistance, that if the maximum light is passing, that means the less resistance, and if the less amount of light is passing, that means the high resistance, right? So based on this concept, this colorimetric biosensor is developed, right? In this case, this is an chemical biosensor is developed where to detect the glucose or starch concentration in a blood sample. This is for checking the glycemic index. What they have done, they have synthesized nanoparticle and alpha amylase nanocomposite, and now when this starch is given as a sample to this nanoparticle, this starch hydrolyzes this alpha amylase, and for that there is a response change in the current. Okay, so this is how this electrochemical. You can go through this paper, then you can understand the uh, principle of this POCD devices. Okay, so this is another LED LDR colorimetric based POCD devices where they are detecting the E. coli bacteria with help of with the help of this LED LDR system. Similar. Next is the detection of micro POCD devices for detection of anti B2 microglobulin, which is present in our urine sample, which is effective for the which can be detected for the diabetes, and it is also related to our vision, right? Eye vision. So when this, so you can see the changes. You can see the device also, and this is the detector. It is also based on this colorimetric assay based principle. Next one is the paper-based alpha amylase detection sensor. So when this, they have taken a filter paper where this starch solution is given with iodine. Now when this alpha amylase is again given, this alpha amylase breaks this starch, right? Alpha amylase breaks the starch, and now it is really, because of the iodine, it is showing some color change, and that is captured in the LDR system. And this things done is completely on a simple paper. So this is the paper-based alpha amylase sensor. Next, a device is formed for calculating the daily blood parameters like heart profile, kidney profile, liver profile, and anemic and diabetic cases, HbA1c, glucose, hemoglobin. With this, applying this UV spectroscopic mechanism, and these devices are already in the final phase of the marketing and checking. So after some days, it will be available in the market for general use. And the last one is another lab has developed a hemoglobin uh, hemoglobin checking device for the anemia detection in the basically it is for the last mile population where there is the device is not available, the laboratory is not available. So what they did, they use a simple paper strip and they are pricking blood from the fingertip, and this blood is given to the test sample strip. Now they are taking the image of the paper strip through this mobile phone, and based on the color change, they have formed an machine learning algorithm, and based on this color thing, they are showing the hemoglobin range, hemoglobin level in the blood. This is a very simple device. Okay, so this is how there are multiple, there are many point of care testing device available, or some are in the development phase also. So you can definitely use various methods, nanotechnology based methods, to develop a new kind of sensor which will be available for human population. So that's it. Thank you for today's class. Thank you for joining. If you have any queries, please uh, comment, or you can raise the question in the discussion forum also. I hope you will find these lectures, all the 4 lectures, because this is the last class, I think. So I think all the 4 lectures will be already, the 3 lectures are available in YouTube. Today's lectures will also be available in the YouTube. If you have any queries, you can comment on the YouTube also, and you can mail me also. And I'll be sharing these PPTs with you. Please go through the lectures. Please go through the slides I have shared, and the YouTube videos I have shared for your reference. And I hope you have understood something from these lectures. So thank you for joining.